So the topic of discussion we are going to have is apoptosis. Okay. So yeah, the topic of discussion we are going to have today is apoptosis. The apoptosis is programmed cell death. It is a suicide program of our cell. Its other name is suicide program. Apoptosis literally means death of in itself. What are the causes found in a cell which led to apoptosis? There may be physiological causes, there may be some causes which are pathological. The first number physiological cause is embryogenesis. When the fetus is developing, we need to cast some cells off to grow more. Okay, during the whole course of development of the fetus, many cells are discarded, and the discarding of those cells by the process of apoptosis. The next thing is endometrial shedding. Endometrial shedding occurs in females during the periods when they are during the menstrual cycle when the decidual shedding or endometrial shedding occurs. That is also a physiological cause of apoptosis. The other is ovarian follicular atresia. I'm not saying ovarian oocyte or germ cell atresia. I am saying follicular atresia, which occurs in females due to lack of hormone supply in their lifespan of when the menopause reach. When there is menstrual cycle, there is occurring of apoptosis in form of endometrial shedding. When there is menopause state, ovarian follicular atresia occurs. These both are example of hormone-related apoptosis. Okay. The next is elimination of abnormal, or I can say immature WBC are lymphocytes in thymus. During the maturation period of our immune system, all the lymphocytes goes through thymus, in which those which are immature or which are not ready to perform the certain activities which a white blood cell should do, that is kill or detect antigen, or some lymphocytes which may self-react on ourselves, which are the main etiology for autoantibodies or autoimmunity disorders. So in thymus, they are all separated out by the, and they also gets degraded by the process of apoptosis. So it's also a physiological cause of apoptosis. The next is the elimination of neutrophils, which is also a WBC. After acute inflammatory response. See, what happens is in an acute inflammatory reaction, there is aggregation of large number of neutrophils. But after that episode, what is the fate of those neutrophils? They have to be degraded because there is no need of them anymore. So these are degraded by the help of apoptosis. It's another example of physiological cause of apoptosis. So the next thing we will discuss is pathological causes. Okay. For pathological causes, which it will related to the mostly abnormal damage in a cell. So going from the center, first is if there is DNA damage, the cell will become abnormal and we want to kill that cell. Okay, we don't need that cell. If a cell got its DNA damage, that cell has to undergo apoptosis. To get a DNA damage, most common things which causes DNA damage are radiation exposure. The next is anti-cancer drugs, which are also cytotoxic drugs. I will write anti-cancer drugs. But to be precise, these are cytotoxic drugs which are toxic to cells. The next is lack of oxygen, that is hypoxia. Hypoxia, we're dealing with. Other two are one from the radiology, oncology department, other is also from oncology department. Our main aspect is hypoxia. Hypoxia can lead to DNA damage, and if the DNA is damaged, the cell will undergo apoptosis. The another pathological DDX to have apoptosis as an example is misfolded protein. See, protein makes the cytoskeletal and all the enzymes in our cell. If these proteins are correctly folded, which is the work of, by the way, chaperones. Chaperones are the from heat shock family whose work is to make folded proteins, and if they do not do their work, then misfolded proteins are made, and misfolded proteins are not functional. So the cell may not function as well. There is a condition when these misfolded proteins get accumulated to a limit where there is a stress on endoplasmic reticulum. That condition is called ER stress. And when this condition arises, the cell will undergo apoptosis. The next condition is if there is an infection of cells under specific infection. Which specific infection I am suggesting? Viruses. In viruses, one example is HIV. If a cell is infected by HIV, then it will be weak. Our body will want it to be apoptosized by pathological means. The other example is any kind of duct obstruction. Duct obstruction will lead to parenchymal atrophy of that gland. If there is any kind of duct obstruction, the parenchyma, where the secretion actually happens, will undergo atrophy, and the cells will die by the process apoptosis. These are the chief causes and examples of apoptosis in physiological and pathological area. Next, we will discuss the morphological changes which occurs during apoptosis. So this is a normal cell. This is a normal cell. This is an apoptotic cell. What are the cellular changes? What are the nuclear changes? We will divide it into two headings. Okay. First is cellular changes. What will happen to the cell size? It will shrink. It will shrink. What will happen to organelles? Organelles, if the cell is shrinking, organelles will be tightly packed. They will come all together. Congested. What happens to cytoplasm? I am saying the cell is shrinking, so it will get dense. And what will be the color we see? Dense pink. And if cell is shrinking, and shrinking, what will happen is there is forces compressing and there will be some outward blebs formation in the cytoplasmic blebs in the cell. We have got cytoplasmic blebs. There will be outpouchings in the cell. These blebs for a chronic period will lead to formation of what? Apoptotic body. Apoptotic bodies are formed in this sequence. First, the cell will shrink, cytoplasm will get dense, organelles will be condensed, organelles will be tightly packed and damage each other. Nuclear, we will discuss the nuclear change, but it will also get compressed, and small packets will be detached from the cell, which are called apoptotic bodies. What will happen to plasma membrane? See, it is a very unique thing, and it is a very characteristic thing that plasma membrane will be intact. It remains intact. Plasma membrane will not be damaged. It may be damaged sometime, but most of the time it is intact. Textbook says it remains intact. We will write it will be intact. Next is nuclear changes. What are the changes occurring inside the nucleus? First is chromatin. We have the chromosome and it is made of chromatin, and chromatin will undergo condensation. Everything is compressing, condensation. Chromatin condensation is most characteristic feature. Most characteristic feature of apoptosis. Where the chromatin condensation occur, if there is a nucleus like this, it will occur in the periphery. In the periphery. I am saying if there is a nucleus, chromatin condensation will occur in the periphery. Location at periphery. In vicinity of, near the nuclear membrane. Okay. There will be dense mass. We will see dense mass in that. What will happen to nucleus as an overall? Nucleus will undergo fragmentation. These are the nuclear changes occurring while the apoptosis is happening. How will the histology of apoptotic cell, how can we recognize it's an apoptosis? The cell we are seeing here is round and oval mass. It is very dense pink. Okay. Dense pink on histological examination, we say it finding like eosinophilic. Eosinophilic, which means pink cytoplasm. Because cytoplasm appears pink. This is pink. The next thing is nuclear changes. We will see fragmented nuclear chromatin. So we are done with the morphological changes of apoptosis. These are very simple. We need to remember cell size shrink, organelle is tightly packed, cytoplasm dense peak, cytoplasmic flap formation of apoptotic bodies, and the plasma membrane remains intact. Nuclear changes occur, chromatin condensation occurs, and nuclear fragmentation occur. Chromatin condensation occur at periphery with formation of dense masses. On histology, round and oval mass is seen, eosinophilic pink cytoplasm is seen, fragmented nuclear chromatin can be observed. Now let us discuss how the actual mechanism, what is the mechanism to occur this whole process of apoptosis, how the cell die. There are mainly two pathways. We will discuss one by one. So first pathway is intrinsic pathway. The second pathway is extrinsic pathway. The first pathway, intrinsic pathway, occurs through the cytoplasmic events and by the organelle, mainly mitochondria. Extrinsic pathway is an immunity related topic. I will discuss both of them. So we got two pathways there. First is intrinsic pathway. We will discuss intrinsic pathway. So there are three things which determines whether the apoptosis will happen or whether the apoptosis will not happen. If there is certain cause, I show you the list of the causes, and it gets sense. If there is embryogenesis or any kind of factor, it gets sense. We need a sensor. The sensors are a particular family which is called as BH3 protein family, and the members are BID, okay, BAD, bad, BIM, NOXA, noxa, PUMA, puma. These you have to remember. Okay, BID, bad, BIM, noxa, puma. See, it's a Hindi trick which I use to remember that is BD is bad, will get you bimar. BD is bad and will get you bimar, and I don't believe in puma stuff. So there is always nuksan in puma because I prefer Adidas and other brands. Okay. So BD is bad and you will get bimar, and there is always nuksan in getting puma. But Virat Kohli will not agree with me. So the next factor is pro-apoptotic. Pro-apoptotic, the factors which wants the cell to die. These are the factors which want cell to die. So these are pro-apoptotic. Any factor which I will write have a code word A in it. Okay. There are basically two we need to remember. That is BAX, BAK. There is A and A in both of them. These are pro-apoptotic. So these factors will be increased when there need to be apoptosis to be done. The next is anti-apoptotic. If there are some pro-apoptotic, there must be some anti-apoptotic which want cell to live longer. What I said is cells which want cell to live longer. So they will have L from the live in themselves. Okay. Any factor I write BCL live. BCL XL live. Next is MCL. So when the apoptosis has to happen, all these factors need to decrease. So BCL, BCLXL, MCL will decrease and BAK increases, then the cell will initiate its apoptosis process. There is one thing I need you to remember, that is growth factors. We got growth factors in our body hormone pathway. They stimulate anti-apoptotic because growth means cell proliferation, that is opposite of cell death. So growth factors will stimulate anti-apoptotic. They want cells to live longer. By the virtue of both these, mitochondrial one phenomena occurs. There is leakage of cytochrome C. Cytochrome C is a important or significant part of ETC pathway. If there is leakage of cytochrome C, we can get an idea there will be no oxidative phosphorylation, there will be no ATP synthesis, and cell will eventually die. So this is the main trigger. The leakage of cytochrome C begins the initiation of cell death by apoptosis. Okay. I will write here leakage of cytochrome C from mitochondria. I have to write it. Mitochondria. This is the correct spelling. What will happen with this cytochrome C? Cytochrome C will come to cytoplasm and combine with a factor called APAF1. That is apoptosis activating factor 1. This is present in cytoplasm and it will combine with cytochrome C, which is leaked from the mitochondria, and form an entity which is called as apoptosome. And this apoptosome will in turn stimulate an enzyme called caspase. And caspase, which caspase? In intrinsic pathways, the caspase which apoptosome stimulate is caspase 9. It's a whole family. Caspase are the enzyme, basically cysteine aspartate protease. The name is cysteine aspartate protease family. These are protein breaking family. Okay, they cleave the protein structure, single line protein structure, after the aspartate amino acid after recognizing the aspartate amino acid. Okay, so our concern is in intrinsic pathways, it will stimulate apoptosome will stimulate caspase number 9. It is an initiator caspase. There are two types of caspase. One is initiator. It will initiate. This is the checkpoint where there is initiation of protein breakdown. Okay, the enzyme is activated. What will execute it? The next is executioner caspase, which will execute the function. These are 3 and 6. Caspase number 3 and caspase number 6 will stimulate the enzymatic activity and breakdown. Okay. What these caspase will do, they do two things. First is endonuclease. You know what endonuclease is? Nucleus is nuclear particle. Endo means in between, and they will cut the genetic material. Endonuclease, whether it is RNA, whether it is DNA, it doesn't matter. Endonuclease activation. So the DNA will break. Another function they have got is protein breakdown. What does protein breakdown mainly consist of? Cytoskeletal damage. Cytoskeletal damage. The whole structural component of a cell which makes the cell unique, okay, which make the cell functional, will be damaged by these. Okay, one thing I left is there are few proteins also there, namely as SMAC and Diablo, which stimulate this execution process. Okay, they stimulate the apoptosis, apoptotic helper. And these are mitochondrial protein actually. They also leak with cytochrome C. SMAC and Diablo. These are in your textbook, okay, so I have to write it. The next is due to the endonuclease activation and protein breakdown, the nucleus will be fragmented and cytoplasm will be dense, and all the small component and blabbing will occur in the cell membrane, and blabbing will occur. A few apoptotic bodies will be formed. Okay. So these two lead to formation of apoptotic bodies. So what if apoptotic bodies is formed? What will occur next? See, if I would have to tell you, this is like a cell membrane. We have learned this in our 12th standard or even in biochemistry first year. This is like a cell membrane. The phosphated inside, the gola is outside. Okay, there will be flipping. What I am saying, there will be flipping. I will write flipping out of phosphatidyl serine. Phosphatidylserine. Why it is important? Flipping out of phosphatidylserine, and this serine is a kind of it's a tasty. It will announce in the cells in our body that please eat me. These signals will be sensed by that I am flipping out. Please, they are showing themselves sensed by macrophage. And other signals are there also. That is one of them is thrombospondin, which is a glycoprotein, which is an adhesive glycoprotein. It will stick to the any cells which come in contact. Other other things can be like complements, any natural or any antibody which sticks to the cell. These will occur. These will act like a opsonization. Okay, they will be sensed by macrophage and they will be attracted to them. And the final step will occur, which is called ephagocytosis by the macrophage. Macrophages will eat up these apoptotic bodies, and that's how the cell death will occur by apoptosis. It's a intrinsic pathway. It's a storyline. We need to go through it again, and it will be done. Next is caspase family. We need to cram this stuff. Caspase family role. They have two roles: initiator and executioner. There are two pathways we got in apoptosis: intrinsic and extrinsic. So initiator we say caspase number 9. Executioner we see it was 3 and 6. I would like to say the initiator is a mitochondrial process. Mitochondria M looks like 3. Everything will be divisible by 3. 3 3 is 9, 3 1 is 3, 3 2 is 6. Okay. The next is and extrinsic pathways. The initiator will change to 8. E8. 8. And executioner is both same, 3 and 6. So there is no change. 3 and 6 is continuous. I would like to tell you that caspase 8 is not human. In human, we find caspase 10. It's a little detail we need to know. In humans, there is caspase 10. That's all about caspase family. Now we will come to extrinsic pathway. See, extrinsic pathway is done by, okay, it's done for self-reacting lymphocytes. It's a part of immune system where a few disarranged immune cells, mainly I am suggesting like self-reactive lymphocytes which react on their own host cells. Okay, these need to be eliminated or they will cause autoimmune disorder or dysfunction in our whole scenario. So what will happen is we have got here T regulatory cells. T regulatory cells. What their function is, they need to recognize self antigens. They recognize self antigens, which are our own antigens. This is a T regulatory cell. These T regulatory cells have a protruding on their surface, which are called TNF, tumor necrotic factor, or another name is fast ligand. Any name is sufficient. Fast protein ligand. And any self-reactive lymphocyte they will bind, they got TNF receptor. TNF receptor have a whole lot family, but the best one we consider is TNF-R1. That is TNF family, receptor family type 1. And other name of TNF receptor will be FAS. FAS is a protein. And this will also known as death receptor. Because if this receptor, if T regulatory cells, TNF or FAS ligand will bind, it will cause the death of this cell. That is, self-reactive lymphocyte will die whenever T regulatory cells come in contact with this cell. Let's say it come in contact with TNF and FAS ligand will contact with TNF receptor or FAS. It will lead to, one second. Let's say TNF or FAS ligand, TNF receptor or FAS, it will come in contact. It will lead to stimulation of a identity and making of FADD, which is called as FAS associated death domain. It will cause FAS associated death domain. That is why another name of extrinsic pathway is death receptor pathway. If this receptor gets stimulated, the cell will die. It's simple as that. And it will stimulated if by T regulatory cell because it detects self antigens, and this cell got self reactive antigens because it will react on self cells, which are our own cells. Okay. FADD will do the same thing which apoptosome did in intrinsic pathway. It will stimulate caspase, but it will stimulate which caspase? We have learned from the family. It will stimulate the initiator caspase, which is caspase 8, and then again executioner caspase, making of apoptotic bodies, and it will be finally engulfed by macrophages. That sequence remains. One protein we need to know that is called flip protein. This inhibits caspase 8. This protein is found in viruses. Few viruses got flipped, they will inhibit extrinsic pathway and they will prevent their own death. See, this is only the mechanism. Now we need to address a simple topic, that is how do you differentiate apoptosis and necrosis. See, apoptosis and necrosis, I already told you, apoptosis is a programmed cell death. It is an active process. It has got a genetic component. That is all true. Necrosis got enzymatic and ischemic process. It is a passive process. We will concentrate on apoptosis because we have read that. Cell size will shrink. Yes, it does. Cells affected. It is a single cell phenomenon because DNA damage, misfolding protein. It is all the causes are inside one cell. Okay, it's not a group event. Okay, the next is nucleus changes. Nucleus changes will be fragmented. Yes, cell membrane will be intact. That is important. Cell membrane will remain intact. There is inflammation. There is no inflammation. It is also important. See, in apoptosis, the cell wants to die on its own. It's a suicide program. Why will there be any other effects? Inflammation effect will not be present in it. The stain we use, the stain is an annexin V. Annexin 5 is the stain to detect apoptosis. And the other thing is gel electrophoresis pattern is step ladder pattern. In necrosis, it is spear pattern. See, cell size is enlarged in necrosis. It will become bigger. Cell affected are grouped because death is in necrosis is a pathological event mostly. Nuclear changes are pycnosis, karyodexis, karyolysis. There will be nucleus will be condensed and it will get fragmented and it will just lyse. Cell membrane will be disrupted. It will be intact. Inflammation will be present. That is these 2 points are very important. Cell membrane and inflammation. The rest are easily can be deciphered by this. Let's see gel DNA gel electrophoresis. Okay. A normal cell, if I do DNA gel electrophoresis, means DNA is present in that medium and DNA will spread across the electric field. So normal cell, there is no DNA damage, there is no acidic component, so it will show just blank. Okay. There will be nothing visible here. In a necrosed cell, all the cell is damaged, membrane is not intact, all the DNA is lysed. DNA is lysed, and it will form a smear. It will uniformly spread throughout the sheet. In apoptosis, there are formation of apoptotic little pouches of nuclear material in them. So it will make a step ladder pattern. These are bigger chunks of nucleic acid present in them. Okay, let's do some questions. Characteristic feature of cytoplasm. Characteristic feature of apoptosis. Cell membrane intact, cytoplasmic eosinophilia, nuclear modeling, cell swelling. Cell swelling is necrosis. Cytoplasmic eosinophilia, yes, it is there, but it is not characteristic. Nuclear molding, I don't know. You don't know, I don't tell you. Cell membrane intact, yet is the answer will be. Which of the following organelles has a pivotal role in apoptosis? Pivotal roles. Pivotal role in apoptosis. Mitochondria, nucleus, endoplasmic reticulum, Golgi apparatus. The answer is mitochondria. All are true regarding apoptosis except the word important is except. So let's check. Chromatin is lysed by endonuclease to form nucleosome. Is a true statement. It initiates local inflammatory reaction. No, it is a false statement. No inflammation in apoptosis. Okay, the answer will be this. Other statements are macrophages, phagocyte remain apoptotic bodies. Yes, that is true. Macrophage does the efferocytosis and engulfs there. Caspases are increased and mediate apoptosis. That's true. They are involved. Next question is apoptotic bodies are clumped chromatin bodies, pycnotic nucleus without cell, without organelles. You know, these are with organelles. Cell membrane bound with organelles. That is the most appropriate. No nucleus with it. No, that is not. Next is annexin V. Annexin 5 is a marker of. It's a marker of apoptosis. Just read it. In apoptosis, APAF1 is activated by release of which of the following substances from the mitochondria? APAF1 is activated by release of which of the following substances from the mitochondria? The answer would be, the answer is cytochrome. The last question is, with the following is an anti-apoptotic gene. Which of the following is anti-apoptotic gene? The answer is, this is apoptotic, this is apoptotic, this is a sensor. The answer will be PCL. This one is anti-apoptotic. So I hope you understand well. This is the module of apoptosis in the topic of cell and its various form of adaptation and death. Thank you for watching.